let's talk about one of the most successful survivors on Earth, Mycobacterium. Now, this isn't just some run-of-the-mill bacteria. We're talking about a whole family of incredibly tough, rod-shaped organisms that are responsible for some of humanity's oldest and most stubborn diseases. So, yeah, let's dive in. So, what is its secret? I mean, how does this tiny organism manage to outsmart our immune systems and survive in conditions that would kill most other bacteria? Well, it all comes down to its very unique, very effective defense system. It's basically a biological suit of armor. And here it is. This is the key to everything. The cell wall of Mycobacterium is absolutely packed with a fatty substance called mycolic acid. We're talking up to 60% of its wall is made of this waxy stuff. You can think of it like a waterproof raincoat, or even better, a suit of armor. This single feature, this is what gives it all its superpowers. And honestly, it's what gives us all our problems. Okay, so let's really break down what this waxy wall actually does. Because it's not just a simple shield, you know? It fundamentally dictates how the bacteria lives, how it grows, and how it defends itself against pretty much everything we can throw at it. What's so wild is how this one single feature has so many different consequences. First off, it makes the bacteria acid-fast. What that means is, once you stain it, it stubbornly holds on to that color even when you wash it with acid, which is a huge clue for identifying it in the lab. Second, that waxy coat is so tough, our own immune cells can swallow it whole and still not be able to destroy it. It also means that standard lab stains, like the gram stain that we use for almost everything else, they just slide right off. And finally, building this incredibly complex, greasy wall takes a ton of energy and time. That's why Mycobacterium is famously slow growing. Some species can take up to 24 hours just to divide once. All right, so we get its defense mechanism. Now let's meet the family. Because the Mycobacterium genus has some real heavy hitters, some of history's most infamous germs, but also some lesser-known relatives just hanging out in the environment. The headliner, of course, is Mycobacterium tuberculosis. This is the one that causes TB. Then you've got Mycobacterium lepra, the ancient cause of leprosy. But there's also this huge, diverse group called the non-tuberculous mycobacteria, or NTMs for short. These guys are everywhere, in the soil, in water, and they're usually totally harmless. But for people with weakened immune systems, they can be really serious opportunistic pathogens. And this table just lays it out so clearly, right? You've got the NTMNs on one side. They're environmental bugs. You get them from exposure to soil or water. The good news is they don't spread from person to person and they're generally weak pathogens. But then you have M. tuberculosis. It has totally adapted to living inside of us. Its home is an infected host. It spreads through the air when people cough, and it is a very effective pathogen. So let's zoom in on the big one, the most significant member of this notorious family, tuberculosis. This disease is a slow, creeping infection that usually goes for the lungs first, but it can actually spread to almost any part of the body. And to get a sense of the scale here, just look at this number. It's estimated that in India, over 40% of the entire population is infected with the TB bacteria. Now, that doesn't mean they're sick. Most are kept in check by a healthy immune system, but it just shows you how incredibly widespread this bug really is. So how does it all start? It all kicks off when someone breathes in tiny airborne droplets from an infected person's cough. Once the bacteria get into the lungs, our frontline immune cells, these things called macrophages, rush in and gobble them up. But here's where that waxy wall becomes a total game changer. The bacteria just live inside the macrophage, totally protected, and start to multiply. The body's only remaining defense is to build a wall around these infected cells, forming a little nodule called a granuloma, or a tubercle, trying to contain the threat. And the disease really has two different phases. Primary TB is that first initial infection. That's when the body forms that little granuloma, called a Gone focus, usually down in the lower parts of the lungs. But the real danger is secondary TB. This is what happens sometimes years later if that dormant infection wakes up and reactivates, maybe because a person's immune system gets weaker. This phase is way more destructive. It creates these big cavities in the lungs, usually up in the very top, and it's when people can become highly contagious. Okay, so how on earth do you find this microscopic intruder? Diagnosing a mycobacterial infection is a lot like detective work, mostly because that waxy wall that makes it so tough also makes it really hard to spot with normal lab tests. The classic tool in the toolbox is a special technique called the Zeal-Nielsen stain. What you do is use heat and a really strong dye to force the color past that waxy wall. Then you try to wash it out with acid. Most bacteria lose the color immediately, but Mycobacterium, it hangs on tight. It resists the acid. It is acid fast. And what's the smoking gun? What does the microbiologist see when they look down the microscope? If it's a positive sample, it's absolutely unmistakable. You see these bright, vivid red rods standing out against a cool blue background of all the other cells. That's your visual confirmation. That's the culprit. But just seeing it sometimes isn't enough. You often need to actually grow it to be sure. And remember, this thing grows at a snail's pace. So labs have to use a special, super nutritious recipe called Lowenstein-Jensen medium, or LJ medium. It's got egg, salts, and this dye called malachite green, which is really clever because it stops all the faster-growing bacteria from taking over the plate. And then you'll wait. And wait. For anywhere from 3 to 8 weeks. After all that patient waiting, what do you finally see on the plate? Well, M. tuberculosis colonies have this very unique look, memorably described as rough, tough, and buff. They're not smooth and shiny like most bacteria. They're dry, crumbly, raised up, and have this kind of creamy, buff color. It's another key piece of evidence for the detective. So we know how to find it in the lab, but now we have to zoom out from the petri dish to the entire planet. Because identifying this bacteria is one thing, but treating it is a massive, ongoing global health challenge, especially because of how tough and slow-growing it is. The main strategy used around the world to fight TB is called DOTS. The logic here is actually pretty simple. The treatment takes months of multiple drugs every single day. It's really easy for patients to stop taking their pills once they start to feel better, but that's dangerous. So DOTS means a healthcare worker literally watches the patient swallow their medicine every day. It ensures they finish the entire course, which is absolutely critical to cure the disease and stop drug resistance from developing. But what happens when the drugs just stop working? That's when we enter the terrifying world of multi-drug-resistant TB, or MDR-TB. These are strains of the bacteria that have evolved to be resistant to the two best, most powerful first-line drugs we have, isoniazid and rifampicin. And that makes treatment incredibly difficult, much more toxic, and way less likely to work. And just when you think it can't get any worse, it does. Extensively drug-resistant TB, or XDR-TB, is the next level up. These strains are resistant not only to those best first-line drugs, but also to the most effective second-line drugs. This leaves doctors with very few and sometimes literally zero effective treatment options. It's a massive threat to global health. So as we pour all of our energy and resources into fighting this global emergency of drug-resistant TB, it leaves us with one final and really important question. With all eyes on the kingpin, M. tuberculosis, are we accidentally overlooking the quiet rise of its environmental cousins, those non-tuberculous mycobacteria, which are increasingly popping up and causing serious disease in vulnerable people all over the world?